Okay, good afternoon. Thank you very much for this kind invitation. It's also good to be here again in the National University. Okay, so I work in Institute for the Study of Inborn Errors of Metabolism in Javeriana University, and I'll share with you some of our results in some of the research lines that we have in our institute about proteins, virus, or vectors for gene therapy, and some small molecules. Okay, so just a brief introduction of this field. We work in inborn errors of metabolism, and I have the same problem with you. Okay, is the microphone? Okay, so we'll try to do it with this. Okay, so these disorders are rare diseases. All of them are monogenic disorders, and they are produced by mutations in genes involved in the but I want to be changing? No. Okay, so these are monogenic disorders, and they are produced by mutations in genes that are involved in the metabolism of amino acids, lipids, carbohydrates, or nucleic acids. So these mutations produce a really wide and variable clinic manifestations and onset age of presentations, and that represents a really challenge for the diagnosis and treatment. And also, most of the drugs for treatment of these disorders are orphan drugs. So in our group, we work in 3 different research lines. One of them is the cellular and molecular basis of these disorders. The other one is the diagnosis. We work in collaboration with Hospital Universitario San Ignacio. And we are one of the centers in Colombia for the diagnosis of these disorders, and we also develop some new tools for the diagnosis of this disease. And also, we have a biotechnology research line for with the idea to develop treatment for the disorder. So I will share with you the results, some of the results from this research line. And just another brief introduction. Among the different research, among the diseases that are in the group of inborn errors of metabolisms, we work especially we work in this group of disorders that are lysosomal storage diseases. Basically, they are a group of diseases produced by mutations in the protein involved in the function of the lysosome. So at the end, that produces an accumulation of substrate, of integrated substrates. And they are progressive disorders and multisystemic disorders affecting several organs, liver disease and nervous system affectation, and other affectation or tissues are affected in these kind of diseases. So one of the main strategies to treat the disorder is the use of recombinant proteins. A strategy called enzyme replacement therapy. Most of the proteins that are used in enzyme replacement therapy are produced in mammalian cells, CHO cells or fibroblasts. We decided to use microorganisms with the idea that we can produce a protein that can be cheaper than the one that is produced in other systems. And we have used E. coli as a system and also yeast to produce some of these proteins. So basically we have produced proteins for Hunter disease, Morquio disease, and GM2 gangliosidosis. Okay, so the first protein that we produced was IDS. It's a protein deficient in Hunter disease, and we use E. coli for that project. So in summary, we were able to produce an active protein. This is a really complex protein, about 500 amino acids, and a lot of protolation and modifications. So we were able to produce the protein with active protein, even with active levels higher than the human leukocytes, but still lower than the CHO cells. But the good thing is that we were able to produce a protein with a kind of processing similar to the one produced with mammalian cells. So this was one of the first studies that we did, and then we observed that this protein has a really interesting stability profile, since it's really high stable to pH 7 with this stability as pH 5, that is a pH of the lysosome. So this will be good if we think in enzyme replacement therapy, since the protein will be infused to the patient. So it will be stable at serum, and this correlates with the stability that we observe of the protein in human serum, which is like higher than the one that is from humans. One of the diseases in which we have put a lot of effort is mercury disease. It's a really common disease in Colombia, although it's also a rare disease. So here we have used first E. coli, and again we were able to produce an active protein here, and we show that E. coli is capable to produce active sulfatases. So we discovered, and we also published the mechanism that is involved in the activation of these human sulfatases in E. coli. And we also noticed that the N-glycosylation are not necessary to produce an active protein in these microorganisms. So we also developed a method to secrete the protein in E. coli, which is not common that these microorganisms secrete protein. So we developed a strategy to produce a secreted protein, and we also observed that this protein has a really good stability, similar to the one produced in mammalian cells, but we were not able that this protein enter to the cell. So because of this problem, we contact this group from Cornell University, from Matthew DeLisa, and we adapted the technology that they developed to produce glycosylated protein in E. coli. So we translated this technology to our lab, and then we produce a glycosylated protein, this protein in E. coli, and we observed that the protein increased the stability, especially at acidic pH, increased the affinity for the substrate, and also was taken up by culture cells, reduced the material that was stored in the cells, and also was located in the lysosome, which is the organelle in which the protein should perform the activity. The other system that we have used is yeast. They are really good microorganisms to produce proteins, especially because the economic culture media that you can use, they secrete the protein with really low contaminants, and also they can produce similar proteolational modification than mammalian cells. Among the different systems that are available, we select Pichia, and we have produced several proteins in this system. So the first one, again, IDS, and we notice here a change in the stability. This protein is more stable at pH 5 and at pH 7. It still is very stable in serum, and this protein, without any further modification, was able to enter to the cells through mannose receptor and was located inside the lysosome. A similar protein, this is another lysosomal protein for a treatment of a disease called GM2 gangliosidosis, Tay-Sachs disease. And here we observe that, again, we can obtain a protein with really high activity, but the good part here is that this protein is able to normalize the lipid storage in fibroblasts and in neural stem cells, so showing that we have a protein that can be translated for therapeutic purposes. Again, our main protein, galans, which is involved in mercury disease. In this case, we also observe a protein that is stable mainly at pH 5, really a good stability, temperature stability is taken out by different cells, Hector-93 cells, fibroblasts, chondrocytes, cardiomyocytes, reduce the material that is undegraded in the cells, but we observe that the yeast produce an hypermanosylation, and that could be a problem if you want to infuse a patient with this protein. So we try to solve this problem. So we did a glycoengineering in the yeast in Pichia pastoris. So for that, we did the knockout of one of the genes involved in the hypermanosylation of the proteins, and also we overexpress some genes involved in the secretion pathway, so we can obtain, we can increase the yield of the protein. And in summary, we obtain a protein with human-like N-glycosylations, and that new glycosylations increase the capture of the protein, so we had a higher amount of protein inside the cells and also in the lysosome, increase the stability and reduce the immunogenicity of the protein. So we are like now doing the experiments with the animal model of the disease, so we can to see if we can normalize or we can treat the disease with this protein. Other field in which we have worked is gene therapy, especially the design of gene therapy vectors. We selected adeno-associated virus vectors, and we started working in this field about year 2000. We were one of the first group in Colombia working in gene therapy, especially using viral vectors. At that time, we need to understand how to produce this virus and to have the techniques and all the protocols for the production and characterizations. Then we move to the evaluation of the promoters, and we compared viral promoters and eukaryotic promoters, and we were able to design an eukaryotic promoter that that produce or express our gene, our protein in similar levels to that using a CMV promoter, but without the side effects that are commonly associated with viral promoters. Then also we evaluate the effect of co-expression of a protein that is important for this kind of enzymes or enzyme of deficient in mercury, that is a sulfatase activator. And we were able to increase significantly the activity and the effects of the gene therapy, and also we performed some in vivo experiments in animal models with the vectors. Nevertheless, here at this point we observe that the vectors, the adeno-associated vectors, they doesn't have a affinity for bond, and this is a bond disease. So we need that the vector reach the bond so we can treat the disease. So to solve that problem, we design a vector with a bond tag peptide in the viral capsid. So the idea was that the vector not only reach the typical tissues that are affected with gene therapy, like liver or mainly a spleen, but also the vector can go straight to bone and in bone express the gene and treat the disease. So just to show you here, this is the biodistribution of the vector. So we have significantly amount of vector in bone and reduction in other tissues that are not important for the disease. And also here we observe a significantly increase of expression of our gene in bone. And also we observe a change in the expression on the biodistribution in other tissues. So the inclusion of this bond tag in the vectors completely changes the biodistribution of the vector. So we think that this vector could be used as a tool to treat other bone diseases that are susceptible to be treated by gene therapy. And just finally, we have also tried to identify small molecules. So to treat this disease, the idea is to use small molecules that can increase the stability or recover the stability of a mutated protein. So at the end, we can recover the the biological function of a mutated protein and treat the disease. In this project, we did a strategy in which we took the 3D structure model of the protein, and we did a computational docking, again, a library of human-approved compounds. And from that, we took the 20 compounds with the highest affinity. We discarded some antibiotics, cytotoxic compounds, estrogen compounds, because the idea is to treat children, so we don't want to use these compounds. And also the idea is to do a daily administration of the product, so we don't want to select one of these products. And when we analyze these compounds inside the protein, we observe that they dock inside the active cavity of the protein in a similar position as that the natural substrates, and they did similar interactions with catalytic residues. And then after a different experimental evaluation, we observed that, in fact, they bind to the active cavity of the protein. They are competed inhibitors. They increase the stability of the protein and increase the activity of recombinant proteins if we add these compounds to the culture of the proteins in yeast, bacteria, or mammalian cells, we significantly increase the production of and activity of the protein. We were able to recover the activity in fibroblasts, chondrocytes, and cardiomyocytes from patients with the disease. And also, we were able to reduce the lysosomal storage. So at the end, we were able to treat the disease with these small molecules by recovering the folding of the protein. We also observed that if we combine our small molecules with the recombinant protein, we have an additive effect of our treatments. So as I know that it's lunchtime, so just a brief conclusions that we have from these years. So we have noted the difference in the glycosylations, just the lack of the glycosylation or the structure of the glycosylations do not prevent the production of active lysosomal enzymes in microorganisms. These enzymes, especially those that are producing yeast or the ones that are producing glycoengineering E. coli, are delivered to lysosomes and then can catalyze the reduction of the storage material. The glycoengineering allows the production of enzymes with homogeneous N glycans that have higher cell uptake. We have the signal and AAV vector that can be tailored to increase their bond biodistribution, and this will be a really nice tool to treat different bond diseases. And also we have identified some pharmacological chaperones that may offer a new alternative for the treatment of Merkel-A disease. In this project, we are now evaluating other diseases that are like harder to treat, such as diseases in which the affected tissue is the central nervous system. So we need to cross the blood-brain barrier that will be not able to treat by using an enzyme. So we are trying to identify molecular chaperones for another disorders. So with this, I think is enough. So thank you very much.